Hello everyone. In this video we are going to discuss about model paper 1 which is given by the PU board exam this year. So until now if you are not subscribed to our channel, subscribe, like and share. Let us start with the first MCQ questions discussion. Statement 1 and statement 2. Pollination does not guarantee the transfer of right type of pollen. So there is pollination where it is not giving a guarantee that there is a right type of pollen is transferred on that. It means that this statement says that pollination takes place, any pollen grain may come and fall on that stigma. So hence this statement 1 is correct. Statement 2. Compatible pollen pistil interaction guarantee double fertilization. So whenever there is compatible pollen, if the right type of pollen will fall on the stigma, then there is pollen pistil interaction, and that one guarantee that there is fertilization, that we call it as double fertilization and triple fusion. Hence both the statements are correct. So the answer will be this one. Both statements 1 and 2 are correct. So next question. The secondary oocyte formation from primary oocyte occurs in which of the follicle? So this is about HR, human reproduction. So secondary oocyte formation takes place from primary oocyte in which of the follicle? So here there are three types of follicles. One is primary follicle, secondary follicle and tertiary follicle. So this is mature Graafian follicle. So here primary follicle is surrounded by follicular cells, and secondary follicle there is theca interna, theca externa differentiation, and tertiary follicle is characterized by antrum cavity, fluid filled cavity, and this is the follicle which releases the secondary oocyte. Hence the answer will be C. So next question. Progenies with A, B and O blood groups are not possible from which of the following mating parents? So here this is not possible. Which one is not possible here they are telling? We should not get AB and O type of blood group progenies. So which one is not possible here? So let us check one by one. First one, AB and O. See, whenever you use this parents like O crossed with AB blood group. So O is small I crossed with AB blood group. Then the progenies will be of A blood group. Then the progenies will be of B blood group only. So here this is the only type of blood group which you will not get AB and O. Hence the answer is A is correct. All others, see already here there is AB. So this A will fuse with this B to form AB blood group. So hence this is compatible. And here also A will mix with B and automatically AB blood group will be there. And here also A with B, AB blood group is there. But this is the only one where it's not possible for the mating parents. Next question. Another chemical name for the nitrogenous base, thymine. You know nitrogenous bases in the DNA, that is purines and pyrimidines. So we have adenine, guanine, cytosine, thymine as well as uracil. So here thymine is a modified uracil, and that modified uracil is 5-methyl uracil. Thymine, another chemical name is 5-methyl uracil. Hence the answer is D. Select the correct match. So here this question is from evolution. That is a diagram where they have given Australian marsupials, placental mammals. We have to identify which one is Australian marsupials called as placental mammal. So spotted cuscus, that is flying squirrel. So actually this is flying phalanger. And bobcat, they have not given here, again Tasmanian bobcat. Then anteater, so this is also wrong. So the correct answer will be spotted cuscus, that is lemur. Lemur is commonly placental mammal where you have Australian marsupial with spotted cuscus difference, or you can say similarity here. So next question. Which of these ancient men protected their body with hide and buried their dead? So here this protection their body and burying their dead is advanced feature. So here the primitive is Dryopithecus, and then comes Homo habilis, then comes Homo erectus, And the last one, Neanderthal man, is the advanced one who used to protect their body and buried their deads, and they are found to be around 1 lakh to 40,000 years back. Hence the answer is B. Next question. HIV, human immunodeficiency virus, replicates in which of the cells? We know that HIV virus first attacks the macrophages and then forms the HIV factory of the cell. So continuously it produces viruses and then it attacks even the helper T cells, and produce more number of these HIV viruses which replicate via reverse transcriptase enzyme. Hence the answer is C. The drug which is a depressant, slows down the body function is. So here we have different types of drugs like opioids, cannabinoids and coca alkaloids. So SMAC is the one which is chemically known as diacetyl morphine, is a depressant that slows down the body function. Hence this is the correct answer. Next question. Saccharomyces cerevisiae, commonly called brewer's yeast, is used for fermenting malted. So in the textbook line they have given malted cereals and fruit juices. So here there is option fruits also, but in the textbook they have mentioned fruit juices. Hence the answer is B here, not C. Next question. In the year 1963, the two enzymes responsible for restricting the growth of the virus bacteriophage in the E. coli were isolated. One of them cut the DNA and the other added the group. So we know in the bacteria, when the virus attacks, its DNA enters inside. Here, to protect the cell, this host DNA will produce two types of enzymes. One will cut the DNA of this and the other one will add the methyl group to the host DNA. So hence the answer will be A, methyl group. Which of the following is the correct representation of the primer annealing step in PCR? So we know polymerase chain reaction. The first step is there is denaturation of the DNA, then annealing process. So and the enzyme which is used in the PCR is, please mention in the comment box. So which enzyme is used in the PCR? It is Tuck polymerase. So this is the enzyme which is active during high temperature. So Tuck polymerase is the enzyme. So here these enzymes, which is a type of DNA polymerase enzyme, always they work from 5' to 3' end. So first we have to find out that one. See, always this opposite, they are 5, 5, so this is wrong, and another one matching wherever 3, 3, that is also wrong. And then you have to find out. So I said Tuck polymerase works from 5' to 3' end. Then the opposite DNA strand should be 3' to 5'. So if you take this one 5' to 3', here it becomes 3' to 5'. Then this is wrong. And this is 3' where from 5' to 3' it runs, and from 5' to 3' it goes on adding nucleotides. Hence the correct option is D. Next question. Given below are the steps in the gene therapy of a patient adenosine deaminase deficiency, where gene therapy. You have studied an example where a 4-year-old girl, she is having this adenosine deaminase deficiency disease, so which causes severe combined immunodeficiency. So here, what are the steps for treatment of gene therapy for that patient? So first step, what you do? So we have to collect lymphocyte cells from the blood of the patients. So here lymphocytes are collected from the blood of the patient. First will be this one. So first we will collect and then we have to grow outside. So this becomes the first one, this is second one. Outside in a culture in the human body. Then we have to add functional ADA gene. So functional ADA gene means this will be third point. And finally, this genetically modified lymphocytes should be transferred into the patient, and this is the fourth one. So So 2, 3, 1, 4. 2, 3, 1, 4. So sorry, 1, we have to say 3, 1, 2, 4. 3, 1, 2, 4. This will be the right answer. So next question. The organisms that reproduce only once in their lifetime. We have different examples here. Only once in their lifetime they will reproduce. So in the plants, can you tell in the plants which type of organism is there, which type of plant is there which will reproduce only once in their lifetime? It is bamboo plant. Only once it will flower and then it will produce large number of fruits and seeds and it will die. So like that, here another one which is producing is salmon fish. Salmon fish in the animals and in the plants, bamboo plant, they are the one who reproduce only once in their lifetime. The decomposers especially abundant in the bottom of the pond are, when we study pond ecosystem, so there is producers and they are the one who catch the sunlight, trap the sunlight and sun energy, convert into chemical energy. After that there is food produced in them and after that their depth is there. So in the depth of the pond there are some decomposers who will decompose the body and then they will again add up their whatever nutrition is there to the water or soil. So like that we have flagellates, fungi and bacteria who are responsible for this one. So they are called decomposers. So some of the students are asking for Kannada version here. I can use some little bit Kannada and then little bit Hindi also. So चलो आगे जाएंगे. Next question क्या है? The IUCN Red List. This is the one which is documenting the extinction of the species in the last 500 years include. So कितना number है वो जो IUCN Red List ने list किया? So here we have different numbers which is jumbled. So here 784 is the extinction of the species in the last 500 years. They have red listed this one. Please by heart this one. It's a numerical type where numbers should be by hearted. So here this is one important question which IUCN Red List has been documented. Naviga fill in the blanks ನೋಡೋಣ ಕನ್ನಡದಲ್ಲಿ ಈಗ. The initial few days of the milk produced after parturition is. So initial few days parturition ಆದ್ಮೇಲೆ produce ಆಗೋ milk ಗೆ ನಾವು ಏನಂತೀವಿ? ಅದಕ್ಕೆ colostrum. So the correct answer is colostrum. Oral pills as contraceptive effectively function. So oral contraceptive ನ ಯೂಸ್ ಮಾಡಿದ್ರೆ ಅದು ಹೇಗೆ ಕೆಲಸ ಮಾಡುತ್ತೆ? So ಅದು ಏನ್ ಮಾಡುತ್ತೆ? ಇನ್ಹಿಬಿಟ್ ಮಾಡುತ್ತೆ ಏನನ್ನ? So here it inhibits ovulation. Symptoms of constipation ಆಗೋದು, abdominal pain ಆಗೋದು, cramps ಆಗೋದು, ಇದೆಲ್ಲ ಯಾವ ಡಿಸೀಸ್ ಗೆ ಸಂಬಂಧ ಇದೆ? So it is related to amoebiasis. Okay, next one. Cyanobacteria is an autotrophic microbe. Cyanobacteria ಏನಿದೆ ಒಂದು ಫುಡ್ ತಯಾರು ಮಾಡುವಂತ ಮೈಕ್ರೋಬ್. ಅದು ಅಕ್ವಾಟಿಕ್ ಮತ್ತು ಟೆರೆಸ್ಟ್ರಿಯಲ್ ಹ್ಯಾಬಿಟೇಟ್ ಒಳಗೆ ನೈಟ್ರೋಜನ್ ನ ಫಿಕ್ಸ್ ಮಾಡುತ್ತೆ. ಅಂತದೊಂದು ಎಕ್ಸಾಂಪಲ್ ಯಾವ್ದು ಇಲ್ಲಿ? ಸೊ ನೈಟ್ರೋಜನ್ ಫಿಕ್ಸ್ ಮಾಡುವಂತದ್ದು ಯಾವ್ದಂದ್ರೆ ಇಟ್ ಈಸ್ nostoc. So this becomes the fourth one, that is 19th one. The last one is Alexander van Humboldt. ಅವನು ಏನ್ ಮಾಡ್ತಾನಂದ್ರೆ ಸ್ಪೀಶೀಸ್ ನ ನೋಡ್ತಾ ಇದ್ದಾನೆ, ಅವನ ಸ್ಪೀಶೀಸ್ ರಿಚ್ನೆಸ್ ಬಗ್ಗೆ ಹೇಳ್ತಾ ಇದ್ದಾನೆ. ಸೊ ಇಟ್ ಇನ್ಕ್ರೀಸ್ಡ್ ವಿತ್ ಇನ್ಕ್ರೀಸಿಂಗ್ ಎಕ್ಸ್ಪ್ಲೋರ್ಡ್ ಏರಿಯಾ ಬಟ್ ಅಪ್ ಟು ಸರ್ಟನ್ ಲಿಮಿಟ್. ಹೇಗೆ ನಾವು ಏರಿಯಾನ ಜಾಸ್ತಿ ಮಾಡ್ಕೋತಾ ಹೋಗ್ತೀವಿ, ಸ್ಪೀಶೀಸ್ ಜಾಸ್ತಿ ಹಾಕೋತಾ ಹೋಗ್ತಾವ ಅಂತ ಹೇಳಿದ್ದು ಅಲೆಕ್ಸಾಂಡರ್ ವ್ಯಾನ್ ಹಂಬೋಲ್ಟ್. ಸೊ ರಿಚ್ನೆಸ್ ಬಗ್ಗೆ ಹೇಳಿದಾನೆ ಅವನು, ಸ್ಪೀಶೀಸ್ ರಿಚ್ನೆಸ್. ದಿಸ್ ವಿಲ್ ಬಿ ದ ಫಿಫ್ತ್ ಒನ್. ಸೊ ಇದು ಕನ್ನಡ ವರ್ಷನ್ ಆಯ್ತು. ನನಗೆ ಕಮೆಂಟ್ ಮಾಡಿ ಕನ್ನಡದಲ್ಲಿ ಚೆನ್ನಾಗಾಗುತ್ತೋ, ಇಂಗ್ಲಿಷ್ ನಲ್ಲಿ ಚೆನ್ನಾಗಾಗುತ್ತೋ ಅಥವಾ ಹಿಂದಿಯಲ್ಲಿ ಚೆನ್ನಾಗಾಗತ್ತೆ ಅಂತ ಹೇಳಿ, ಸೊ ದಟ್ ಅಪ್ಕಮಿಂಗ್ ವಿಡಿಯೋಸ್ ಅನ್ನ ಅದರಲ್ಲಿ ಮಾಡೋಣ. ನೆಕ್ಸ್ಟ್, ಸೊ ನೌ ವಿ ಗೋ ಟು 2 mark questions. Draw a neat label diagram of mature 2-celled pollen grain. See, we have already given a blueprint video where how much marks is for each chapter allotted and what type of questions is framed, either 2 mark, 3 mark or 5 mark. Even passing package hakidvi. You just go through that, and this is one of the important question. You have to draw the diagram with exine. If possible, this you can label it as exine, and this you can label it as intine, and then the larger cell is called vegetative cell and the smaller one is called as generative cell. Next question. 4 assisted reproductive technique, mention them, and you can advise them for infertility problems. So ಹಾಗಾದ್ರೆ ಯಾವ ಇದಾವು? Which are the 4 assisted reproductive technique? So we have more than that. So you can tell in vitro fertilization, embryo transfer, or zygote intrafallopian transfer, or gift, IUT. So you can mention any 4 from this. Recombinants obtained in both Mendelian and Morgan experiment. Mendel also did the experiment and Morgan also did the experiment. Both are recombinants, but yet there is something, there is different on that. Process is different. Then what is that? See, Mendel selected 7 contrasting characters, but luckily these all characters were present on different chromosomes. Sorry students, we had a small disturbance here when the card is full, so we replaced, as there is a small cut. Adjust with that. So recombinants with the Mendel and Morgan experiment are different because Mendel characters what was selected for the experiment was present on different chromosomes, but Morgan they were present on the same chromosomes and they were linked with each other. So there is exchange between non-sister chromatids of homologous chromosome, hence they are linked, but these are not linked. Next question. Name the methodologies used in HGP, human genome sequencing or human genome project. So there are 2 methods. One is simply sequencing the whole DNA. The other one is only the DNA which express. So hence the DNA which express is expressed sequence tags. The other method is sequence annotation. Simply the whole DNA is annotated or it is detected. Next question. Describe briefly the primary treatment of sewage. So we have 2 steps in the sewage treatment, primary and secondary treatment. Primary treatment involves physical removal of particles. It may be large or it may be small sized from the sewage, and the steps involve filtration and sedimentation. So filtration, initially it is floating debris is removed, and sedimentation removes soil and small pebbles which we call grit, and that is by sedimentation process. Next question. Name the evolution of any 2 plant forms. 2 plant forms through geological periods from Tracheophyte ancestors. When you see the chart of the zoological time periods, Cenozoic era, Mesozoic era, there you come across Tracheophyte ancestors. So you can mention Zoosterophyllum, herbaceous lycopods, arborescent lycopods. The meaning of this is zoological time scale ಕೊಟ್ಟಿದ್ದಾರೆ, ಅದರೊಳಗೆ ಟ್ರಾಕ್ಯೋಫೈಟ್ ಇಂದ ಬಂದಿರುವಂತ ಆನ್ಸೆಸ್ಟರ್ಸ್ ಯಾವ್ಯಾವ ಇದಾವೆ ಅಂತ ಮೆನ್ಷನ್ ಮಾಡಬೇಕು. ಆ ಚಾರ್ಟ್ ಅಲ್ಲಿ ಕೊಟ್ಟಿರೋದು ಎಕ್ಸಾಂಪಲ್ಸ್ ಇವು, ಸೊ ಅದನ್ನ ನೀವು ಇದು ಮಾಡಬಹುದು. Differentiate between ex situ and in situ conservation. So in situ conservation, so natural habitat ಒಳಗೆ ಆ ಸ್ಪೀಶೀಸ್ ನ ಅಲ್ಲೇ ಬಿಟ್ಬಿಡ್ತೀವಿ. So conservation of endangered or threatened species in their natural habitat. Example is national parks, biosphere reserves, wildlife sanctuaries. Ex situ conservation, so here the endangered species or threatened animals or plants, they are left in natural habitat. So that is away from the natural habitat, it is in the natural habitat. So ex situ, away from the, means outside the natural habitat, in situ, in the natural habitat. So it includes botanical garden, zoo, cryopreservation, seed or pollen banks or gene banks. You can give one definition and then 1 or 2 examples. Next question. Now we come to part C for 3 marks. Draw neat label diagram of monocot embryo. So you have to practice this diagram and label. 3 means either you have to label 6 labelings. So coleorhiza, root cap, radicle, then epiblast, shoot apex, coleoptile and scutellum. So practice this diagram. What is menstrual cycle? The reproductive cycle in the female primates, that is monkeys, apes and human beings, it is called as menstrual cycle. So here in these female primates, there is a reproductive cycle. In the non-female primates, it is called as estrous cycle, but in the female primates, it is called as menstrual cycle. Next part of the question. Mention one pituitary hormone. Pituitary hormone, FSH and LH. Follicle stimulating hormone, luteinizing hormone. Among these, you can mention one. Ovarian hormone. Ovarian hormone is estrogen and progesterone. Again, you have to mention any one from that. Write any 3 principles to be followed by people to be free from sexually transmitted infections. So you have to tell 3 principles to avoid sexually transmitted infections. So what thing you can do is avoid the sex with unknown or multiple partners. Then always try to use condoms during coitus. In case of doubt, one should go to a qualified doctor for early detection and take a complete treatment for the diagnosis, whatever infection is there. Draw a neat label diagram of Stanley-Miller's experiment in relation to chemical origin of life. So this diagram is very important from this evolution point. So practice this diagram and label the parts. Name the causative organism of the following disease. First one is filariasis. Filariasis is caused by Wucheria bancrofti or Wucheria malayi. Ringworm disease is caused by a fungi, Microsporum, Trichophyton or Epidermophyton. Malaria is caused by a protozoa called Plasmodium, Plasmodium vivax or malaria or falciparum. Mention the uses of following bioactive agents. Streptokinase is used as clot buster. So for removing clots from the blood vessels of the patients who are suffering from myocardial infarction leading to heart attack. Heart attack ಯಾರಿಗಿರುತ್ತೆ ಅವ್ರಿಗೆ ನಾವು ಸ್ಟ್ರೆಪ್ಟೋಕೈನಸ್ ಕೊಟ್ರೆ ಆ ಬ್ಲಡ್ ಅಲ್ಲಿ ಕ್ಲಾಟ್ ಆಗಿತ್ತಂದ್ರೆ ಆ ಬ್ಲಡ್ ಅದೇನಾಗುತ್ತೆ ಬರ್ಸ್ಟ್ ಆಗುತ್ತೆ. ಸೊ ಸ್ಟ್ರೆಪ್ಟೋಕೈನಸ್ ಅದಕ್ಕೆ ಯೂಸ್ ಮಾಡ್ತೀವಿ. ನೆಕ್ಸ್ಟ್ ಸೈಕ್ಲೋಸ್ಪೋರಿನ್ ಎ ಅದನ್ನ ಯಾಕೆ ಯೂಸ್ ಮಾಡಬೇಕು? It is used as immunosuppressive agent during organ transplant patients. Organ ಅನ್ನ ಕಿಡ್ನಿ ಫೇಲ್ ಆಗಿದ್ರೆ ಕಿಡ್ನಿ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡಿದ್ವಿ ಅಂದ್ರೆ ಆರ್ಗನ್ ಅನ್ನ ಟ್ರಾನ್ಸ್ಪ್ಲಾಂಟ್ ಮಾಡಿದ್ವಿ ಅಂದ್ರೆ ಅವಾಗ ಬಾಡಿ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಆಟೋ ಇಮ್ಯುನಿಟಿ ಆಗುತ್ತೆ. ಸೊ ಅಂತ ಟೈಮ್ದಲ್ಲಿ ನಾವು ಇಮ್ಯುನೋಸಪ್ರೆಸಿವ್ ಏಜೆಂಟ್ ಯೂಸ್ ಮಾಡಬೇಕಾಗತ್ತೆ. ಅದು ಸೈಕ್ಲೋಸ್ಪೋರಿನ್ ಎ ಅಂತ ಯೂಸ್ ಮಾಡಬಹುದು. ಸೊ ಸ್ಟಾಟಿನ್ಸ್ ಆರ್ ಯೂಸ್ಡ್ ಆಸ್ ಬ್ಲಡ್ ಕೊಲೆಸ್ಟ್ರಾಲ್ ಲೋವರಿಂಗ್ ಏಜೆಂಟ್ಸ್. ಬ್ಲಡ್ ಅಲ್ಲಿ ಕೊಲೆಸ್ಟ್ರಾಲ್ ಜಾಸ್ತಿ ಆಗಿತ್ತಂದ್ರೆ ಸ್ಟಾಟಿನ್ಸ್ ಅನ್ನ ಕೊಡ್ತಾರೆ. ದೇ ಆರ್ ಅವೈಲಬಲ್ ಇನ್ ದ ಫಾರ್ಮ್ ಆಫ್ ಮೆಡಿಸಿನ್ಸ್. ಸೊ ಅದೇನ್ ಮಾಡುತ್ತೆ ಅಂದ್ರೆ ಬ್ಲಡ್ ಅನ್ನ ಕೊಲೆಸ್ಟ್ರಾಲ್ ಅದರೊಳಗಿರೋದನ್ನ ಲೋವರಿಂಗ್ ಮಾಡುತ್ತೆ, ಕಡಿಮೆ ಮಾಡುತ್ತೆ. ಡಿಸ್ಕ್ರೈಬ್ ದ ಸ್ಟೆಪ್ಸ್ ಆಫ್ ಡಿಕಾಂಪೋಸಿಷನ್ ದಟ್ ಆಪರೇಟ್ ಸೈಮಲ್ಟೇನಿಯಸ್ಲಿ ಆನ್ ದ ಡೆಟ್ರಿಟಸ್. ಸೈಮಲ್ಟೇನಿಯಸ್ ಆಗಿ ಆಕ್ಟ್ ಮಾಡುವಂತ ಡೆಟ್ರಿಟಸ್ ಮೇಲೆ ಸ್ಟೆಪ್ ಯಾವಿದಾವೆ ಡಿಕಾಂಪೋಸಿಷನ್? ಸೊ ಇಂಪಾರ್ಟೆಂಟ್ ಸ್ಟೆಪ್ ಸೈಮಲ್ಟೇನಿಯಸ್ಲಿ ಆಕ್ಟ್ ಮಾಡಬೇಕಾದ್ರೆ ಫ್ರಾಗ್ಮೆಂಟೇಶನ್, ಲೀಚಿಂಗ್, ಕೆಟಬಾಲಿಸಮ್ ಇವು ಮೂರು ಅಟ್ ಎ ಟೈಮ್ ನಡೀತಾ ಇರ್ತಾವ. ಸೊ ಹಾಗಾಗಿ ಆ ಡೆಡ್ ಅದು ಲೀಫ್ ಇರ್ಬೋದು ಅಥವಾ ಅನಿಮಲ್ ಇರ್ಬೋದು ಅಥವಾ ಯಾವ್ದೋ ಡೆಬ್ರಿಸ್ ಇರ್ಬೋದು, ಅದ್ರ ಮೇಲೆ ಅದು ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಈ ಮೂರು ಸ್ಟೆಪ್ ಅಟ್ ಎ ಟೈಮ್ ನಡೀತಾ ಇರ್ತಾವ. ಸೊ ಫಸ್ಟ್ ಡೆಟ್ರಿಟಿವೋರ್ಸ್ ಲೈಕ್ ಅರ್ಥವರ್ಮ್ ಏನ್ ಮಾಡ್ತಾವ ಅಂದ್ರೆ ಫ್ರಾಗ್ಮೆಂಟೇಶನ್ ಮಾಡ್ತಾವ. ದೇ ವಿಲ್ ಬ್ರೇಕ್ ಡೌನ್ ದ ಡೆಟ್ರಿಟಿಸ್ ಇಂಟು ಸ್ಮಾಲರ್ ಪಾರ್ಟಿಕಲ್ಸ್. ಅದೇ ತರ ನೆಕ್ಸ್ಟ್ ಲೀಚಿಂಗ್. ಈ ಟೈಮಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ವಾಟರ್ ಸಾಲ್ಯುಬಲ್ ಇನ್ಆರ್ಗ್ಯಾನಿಕ್ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಇದ್ರೆ ದೇ ಗೋ ಡೌನ್ ಇನ್ ಟು ದ ಸಾಯಿಲ್ ಅಂಡ್ they will precipitate and become unavailable salts. The next, catabolism. Bacteria, fungi, enzymes. They will produce enzymes and they degrade the detritus into simple inorganic substances. So you move at a time step nadiyodinda. So they have asked, describe which one operates simultaneously on the detritus. So we come to 5-mark question. Draw a neat label diagram of human male reproductive system. Hope you all know that this is very important question. HR bandre, human reproduction bandre, male reproductive system, female reproductive system, then structure of sperm, mattu we have mammary gland. ಅದೆಲ್ಲ ಏನಂದ್ರೆ ಇಂಪಾರ್ಟೆಂಟ್ ಡಯಾಗ್ರಾಮ್ಸ್ ಇದಾವು. So everyone should practice that, and this diagram also is very important for labeling. But this question, you see, diagram you have to draw, and they have given the labelings, you have to label only that. So they have given testis, some anal vesicles, then glans penis, epididymis, urethra. So now ಎಲ್ಲ diagram ಅಲ್ಲಿ ಎಲ್ಲ ಅಷ್ಟು labeling ಕೊಟ್ಬಿಟ್ಟಿದ್ವಿ, ನೀವು ಇವಿಷ್ಟೇ labeling ಮಾಡಬೇಕು. So schematically represent two gene interaction by using seed shape as well as seed color. So you already know that seed shape ಯಾವ್ದಿದೆ. So it is round and wrinkled, color yellow and green. So capital R, capital Y, round yellow seeds crossed with wrinkled green seeds. So gametes formation is there. You can go with step by step. First parents phenotype, then parents genotype, and then gametes, and then F1. So this becomes parents phenotype. This is parent phenotype, and this is parent genotype, and this is gamete, and then you get F1 generation. Capital R, capital Y, crossed with small r, y, you get capital R, capital Y, small y. In the F2 generation, you get the 9:3:3:1 as a phenotypic ratio, and the genotypic ratio is not necessary to write as it is not given in the textbook. You can write only the phenotypic ratio. DNA is genetic material in both prokaryotes and eukaryotes, yet the packaging process in their cells is not same. Explain. See, we have genetic material in prokaryotes also, eukaryote also. Prokaryote and eukaryote, both the DNA, there is a package, and this packing is different. It is not same. So explain. So you have to use here nucleosome model in the eukaryotes. In the prokaryotes, you can tell they don't have defined nucleus because the DNA is not scattered throughout the cell. It is held with small or some proteins which are positive charges, and it is called as nucleoid, and this nucleoid is organized in large loop held by proteins. So this will be the prokaryote part, and in the eukaryotes what happened? Here much more complex is there. There is a set of positively charged basic proteins, we call them as histones. And how do these protein acquire charge? It depends upon the abundance of amino acids and with their charged side chain, and these histones are rich in basic amino acids like lysine, arginine. You can use these points. Both the amino acid residues carries their positive charges in the side chains. So histones are made up of a unit of 8 molecules. They are called as histone octamer, and negatively charged DNA will coil. This is histone protein. Negatively charged DNA will coil around the positively charged protein like this, and it form a structure called as nucleosome. Hence, this nucleosome contain how much DNA base pairs? 200 base pairs. So continuously what happened? The DNA coil and become more number of nucleosomes, and this can be observed as nucleosome, that is bodies in the nucleus, stained structures. The nucleosomes in chromatin appears like beads on the string structure in the electron microscope, and hence it is also called as beads on string model, and further coil and they become chromosome during metaphase stage. So even the packing of this chromatin at the higher level requires a set of proteins which are called as non-histone chromosomal proteins. So this much points you can write. Go for the next question. Differentiate between innate immunity and acquired immunity. Innate immunity is a immunity which we get by birth. Acquired, it is after the birth, during the growth. So this innate is a non-specific type of defense. It can kill any organism. But acquired develops when a pathogen encounters the host body. ನಮ್ಮ ಬಾಡಿ ಒಳಗಡೆ ಬಂದಾಗ ಪ್ಯಾಥೋಜನ್ ಅವಾಗೇ ನಮಗೆ ಏನು ಇಮ್ಯುನಿಟಿ ಸಿಗುತ್ತಲ್ಲ ಅದು ಅಕ್ವೈರ್ಡ್. ಹುಟ್ಟಿದಾಗಿಂದ ಬರುವಂತ ಇಮ್ಯುನಿಟಿಗೆ ನಾವು ಇನ್ನೇಟ್ ಇಮ್ಯುನಿಟಿ ಅಂತೀವಿ. ಸೊ ಹಾಗಾಗಿ ಇಟ್ ಈಸ್ ಪ್ರೆಸೆಂಟ್ ಅಟ್ ದ ಟೈಮ್ ಆಫ್ ಬರ್ತ್, ಇಟ್ ಈಸ್ ಪ್ಯಾಥೋಜನ್. it is afterwards. Then it is not characterized by memory. ಇಲ್ಲಿ ಬೈ ಬರ್ತ್ ಇಂದ ಬರೋದು ಮೆಮರಿ ಇರಲ್ಲ, ಬಿಕಾಸ್ ವೈಡ್ ರೇಂಜ್ ಆಫ್ ಪ್ಯಾಥೋಜನ್ಸ್ ಗೆ ಅದು ಅಟ್ಯಾಕ್ ಮಾಡುತ್ತೆ. ಬಟ್ ಅಕ್ವೈರ್ಡ್ ಏನಂದ್ರೆ ಮೆಮರಿ ಇರುತ್ತೆ. ಸೊ ಕ್ಯಾರೆಕ್ಟರೈಸ್ ಬೈ ಮೆಮರಿ. ದೆನ್ ಇಟ್ ಪ್ರೊವೈಡ್ಸ್ ಇಮ್ಯುನಿಟಿ ಆರ್ ಪ್ರೊಟೆಕ್ಷನ್ ಥ್ರೂ ಡಿಫರೆಂಟ್ ಟೈಪ್ಸ್ ಆಫ್ ಬ್ಯಾರಿಯರ್ಸ್. ಬಾಡಿ ಒಳಗೆ ಬ್ಯಾರಿಯರ್ಸ್ ಇರ್ತವೆ, ಆ ಬ್ಯಾರಿಯರ್ಸ್ ಥ್ರೂ ನಮಗೆ ಪ್ರೊಟೆಕ್ಷನ್ ಕೊಡುತ್ತೆ, ಇನ್ನೇಟ್ ಇಮ್ಯುನಿಟಿ. ಬಟ್ ಅಕ್ವೈರ್ಡ್ ಇಮ್ಯುನಿಟಿ ಪ್ರೊವೈಡ್ಸ್ ಇಮ್ಯುನಿಟಿ ಥ್ರೂ ದ ಪ್ರೊಡಕ್ಷನ್ ಆಫ್ ಆಂಟಿಬಾಡೀಸ್. ಆಂಟಿಬಾಡೀಸ್ ಬಿ ಲಿಂಫೋಸೈಟ್ಸ್ ಇರ್ಬೋದು, ಟಿ ಲಿಂಫೋಸೈಟ್ಸ್ ಅಲ್ಲಿ ತಯಾರಾಗ್ತವೆ. ಸೊ ಯು ಕ್ಯಾನ್ ಟೇಕ್ ದಿಸ್ ಡಿಫರೆನ್ಸಸ್. Mention the cells involved in accomplishing the acquired immunity. Just now ಮಾತಾಡಿದ್ವಿ ನಾವು ಬಿ ಲಿಂಫೋಸೈಟ್ಸ್ ಮತ್ತು ಟಿ ಲಿಂಫೋಸೈಟ್ ಸೆಲ್ಸ್, ಅವೇ ನಮಗೆ ಅಕ್ವೈರ್ಡ್ ಇಮ್ಯುನಿಟಿಯನ್ನು ಕೊಡೋದು. ಸೊ ಡಯಾಗ್ರಾಮ್ ಕೊಟ್ಟಿದ್ದಾರೆ. ನೇಮ್ ದ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಟ್ರಾಪ್ಡ್ ಇನ್ ದ ಲಿಂಫ್ ನೋಡ್ಸ್ ರೆಸ್ಪಾನ್ಸಿಬಲ್ ಫಾರ್ ದ ಆಕ್ಟಿವೇಶನ್ ಆಫ್ ಲಿಂಫೋಸೈಟ್ಸ್ ದಟ್ ಬೈಂಡ್ ಟು ಎ ಅಂಡ್ ಬಿ ಸೈಟ್. ಇಲ್ಲಿ ಎ ಮತ್ತು ಬಿ ಸೈಟ್ ಇದೆ. ಸೊ ಅಲ್ಲಿ ಏನ್ ಬೈಂಡ್ ಆಗುತ್ತೆ? ಸೊ ದೇರ್ ಇಟ್ ಈಸ್ ಅ ಪಾರ್ಟ್ ವೇರ್ ಮೈಕ್ರೋ ಆರ್ಗಾನಿಸಮ್ ಬೈಂಡ್ಸ್, actually antigen binding site antive adike. So what does H2L2 represent? H2L2 andre enu? It is nothing but light chains and heavy chains anta karitivi now. So again we had a small technical issue. So let's continue with 39 question A. Explain the process of preparation of competent host for transformation with rDNA. So this is A question and B question is there. So here we use, if you want to transfer the rDNA into the host cell, first we have to treat with a specific divalent cation, that is calcium. And this, if you do, it increases the efficiency of the DNA into the bacteria through pores, where in the cell wall. So rDNA, afterwards you can force them into the cells by incubating with rDNA on the ice, followed by again briefly you have to give 42-degree heat shock and then again put them back on the ice. So the bacteria will take up the rDNA. So the next B, refer to the given diagram of PBr 322. Answer the following question that follows. They are given the diagram. Mention the restriction enzymes that can be used for insertional inactivation of antibiotic resistant genes. So we have two antibiotic resistant gene. One is ampicillin resistant, the other one is tetracycline resistant. So in that area, what type of restriction enzymes can be used? So we can use PVU1, PST1 in the ampicillin resistant. In the tetracycline resistant, you can use BUM-H1 and SAL1. So here they have got BUM-H1 as well as SAL1. How many cloning sites are present in the given PBr 322? Cloning sites. So you have to count this. 1, 2, 3, 4, 5, 6, 7, 8 cloning sites are there. So you can mention them. Genetically modified plants have been developed through core techniques of biotechnology. So how are these genetically modified plants useful to humans? So now we have to tell these genetically modified plants how they are useful for us. So these crops are more tolerant. So which type of organisms are genetically modified? They may be plants, they may be animals, or they may be fungi or bacteria. So here plants we are speaking. So these plants are made tolerant to abiotic stress like cold, drought, salt, heat. ಸೊ ಈ ಪ್ಲಾಂಟ್ಸ್ ಅನ್ನು ನಾವು ಏನ್ ಮಾಡಬಹುದು? ಜೆನೆಟಿಕಲಿ ಮಾಡಿಫೈ ಮಾಡಿದ್ಮೇಲೆ ಈ ಪ್ಲಾಂಟ್ಸ್ ಟಾಲರೇಟ್ ಮಾಡೋ ಕೆಪಾಸಿಟಿ ಇರುತ್ತೆ ಸ್ಟ್ರೆಸ್ ಅನ್ನ. ಕೋಲ್ಡ್, ಬಹಳಷ್ಟು ಕೋಲ್ಡ್ ಇದ್ರೂ ದೇ ಸರ್ವೈವ್, ಡ್ರೌಟ್ ಇದ್ರು ಸರ್ವೈವ್, ಸಾಲ್ಟ್ ಇರ್ಲಿ, ಹೀಟ್ ಇರ್ಲಿ, ಎಲ್ಲವಕ್ಕೂ ಸರ್ವೈವ್ ಆಗೋದಕ್ಕೆ ಇದನ್ನ ನಾವು ಜೆನೆಟಿಕಲಿ ಮಾಡಿಫೈ ಪ್ಲಾಂಟ್ಸ್ ಅನ್ನ ಮಾಡ್ತೀವಿ. ಸೊ ಲೈಕ್ ದಟ್ ರೆಡ್ಯೂಸ್ ರಿಲಯನ್ಸ್ ಆನ್ ಕೆಮಿಕಲ್ ಪೆಸ್ಟಿಸೈಡ್ಸ್. ಸೊ ಈಗ ಏನದು ಪೆಸ್ಟ್ ಅಟ್ಯಾಕ್ ಆಯ್ತಂದ್ರೆ ಸೊ ನಾವು ಪೆಸ್ಟಿಸೈಡ್ ಹೊಡೆಯೋದು ಬೇಕಾಗಲ್ಲ, ಸಿಂಪಡಿಸೋದು ಬೇಕಾಗಲ್ಲ, ಬಿಕಾಸ್ ದೇ ಆರ್ ಮೇಡ್ ಪೆಸ್ಟ್ ರೆಸಿಸ್ಟೆಂಟ್. ಸೊ ನಾವು ಅದ್ರ ಮೇಲೆ ರಿಲಯನ್ಸ್ ಆಗೋದು ಬೇಕಾಗಲ್ಲ, ಡಿಪೆಂಡ್ ಆಗೋದು ಬೇಕಾಗಲ್ಲ. ಹೆಲ್ಪ್ ಟು ರೆಡ್ಯೂಸ್ ಪೋಸ್ಟ್ ಹಾರ್ವೆಸ್ಟ್ ಲಾಸ್. ಸೊ ಆಫ್ಟರ್ ದ ಪ್ರೊಡಕ್ಷನ್ ಆಫ್ ದ ಫ್ರೂಟ್ಸ್ ಆರ್ ಎನಿಥಿಂಗ್ ಹಾರ್ವೆಸ್ಟಿಂಗ್, ಸೊ ಲಾಸ್ ಆಗ್ಲಾರ್ದಂಗೆ ನಾವು ಈ ಪ್ಲಾಂಟ್ಸ್ ಅನ್ನ ಇದು ಮಾಡಬಹುದು. ಸೊ ದೇ ಆರ್ ಡೆವಲಪ್ಡ್ ಫಾರ್ ದಟ್. ಇನ್ಕ್ರೀಸ್ ಎಫಿಷಿಯನ್ಸಿ ಆಫ್ ಮಿನರಲ್ ಯೂಸೇಜ್ ಬೈ ಪ್ಲಾಂಟ್ಸ್. ಮಿನರಲ್ ಸಾಯಿಲ್ ಒಳಗಿನ ಮಿನರಲ್ಸ್ ನ ಎಫಿಷಿಯನ್ಸಿಯಾಗಿ ಅದು ಇನ್ಕ್ರೀಸ್ ಮಾಡುತ್ತೆ ಅಬ್ಸಾರ್ಪ್ಷನ್ ನ. ಹಾಗಾಗಿ it will not absorb all the nutrients from the soil. It absorbs which minerals are required, only that one, so that it can prevent the early exhaustion of fertility of soil. Then these genetically modified plants are produced to enhance the nutritional value of food. The best example is golden rice. Golden rice ಅಲ್ಲಿ ಏನಿರುತ್ತೆ, vitamin A ಜಾಸ್ತಿ ಇರುತ್ತೆ, N rich ಆಗಿರೋದು. So these are some answers you can write, points can be written. Write an equation to calculate the population density at the time t. Population density n given time. So it will be nt plus. So natality, this is natality, that is birth plus immigration, and death plus immigration. So this is mortality plus immigration. So you can give this equation. So we have A, B here. B question. Population density fluctuates due to change in 4 basic process. Population ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಫ್ಲಕ್ಚುವೇಟ್ ಆಗುತ್ತೆ, ಹೆಚ್ಚು ಕಡಿಮೆ ಆಗುತ್ತೆ. 4 ಬೇಸಿಕ್ ಪ್ರೋಸೆಸ್ ಇಂದ ಮೆನ್ಷನ್ ಮಾಡ್ಬೇಕು ಅದನ್ನ ಮತ್ತು ಡಿಫೈನ್ ಮಾಡ್ರಿ ಅಂತ ಹೇಳಿದ್ದಾರೆ. So factors responsible for fluctuating population density are natality, mortality, immigration and emigration. Natality means number of births given period in a population. So ಜಾಸ್ತಿ ಮಕ್ಕಳು ಹುಟ್ಟಿದ್ವು ಅಂದ್ರೆ ಆಟೋಮ್ಯಾಟಿಕ್ಲಿ ಪಾಪ್ಯುಲೇಷನ್ ಡೆನ್ಸಿಟಿ ಜಾಸ್ತಿ ಆಗುತ್ತೆ, ಅದು ನಟಾಲಿಟಿ. ಮಾರ್ಟಾಲಿಟಿ, ಡೆತ್ ಜಾಸ್ತಿ ಆದ್ರೆ ಇನ್ ಅ ಗಿವನ್ ಪೀರಿಯಡ್ ಆಫ್ ಟೈಮ್ ಡಿಕ್ರೀಸ್ ಆಗುತ್ತೆ ಪಾಪ್ಯುಲೇಷನ್. ಇಮ್ಮೈಗ್ರೇಷನ್, ನಂಬರ್ ಆಫ್ ದ ಇಂಡಿವಿಜುವಲ್ ಆಫ್ ದ ಸೇಮ್ ಸ್ಪೀಶೀಸ್ that have come into the habitat. ಈಗ ಬೇರೆ ದೇಶ ಬಿಟ್ಟು ನಮ್ಮ ದೇಶದಲ್ಲಿ ಬಂದು ಸಿಟಿಜನ್ಶಿಪ್ ತಗೊಂಡು ಇದ್ರಂದ್ರೆ ಅದು ಇಮ್ಮೈಗ್ರೇಷನ್, ಪಾಪ್ಯುಲೇಷನ್ ಆಟೋಮೆಟಿಕ್ಲಿ ಇನ್ಕ್ರೀಸ್ ಆಗುತ್ತೆ. ಸೊ ಈ ಮೈಗ್ರೇಷನ್ ನಮ್ಮ ದೇಶ ಬಿಟ್ಟು ಬೇರೆ ದೇಶಕ್ಕೆ ಹೋದ್ರೆ ಇಟ್ ಈಸ್ ದ ನಂಬರ್ ಆಫ್ ಇಂಡಿವಿಜುವಲ್ಸ್ ಆಫ್ ದ ಪಾಪ್ಯುಲೇಷನ್ left the habitat, so or elsewhere in the given period of time under consideration ತಗೊಂಡ್ರೆ that is immigration. These are the main 4 basic process. Complete the given tabular column with respect to male and female gametophyte of angiosperms of the mentioned character. So 42 question na sexual reproduction in flowering plants question ide. They have given this one, you have to fill this one. So here number of cells present in the mature male gametophyte. So either it may be 2-celled or 3-celled. Adhe tara female gametophyte idre, it will be 7-celled, 8 nucleated ide. Number of gametes. So in the pollen grain, you can see from the pollen grain there is vegetative cell, generative cell. Generative cell will produce 2 gametes. So here number of gametes are 2 gametes, and female gametophyte ali there is only 1 egg cell. Type of cell division involved in during the development. Development ಆಗಬೇಕಾದ್ರೆ ಮೊದಲು meiosis ಆಗುತ್ತೆ, ಆಮೇಲೆ development ಆಗಬೇಕಾದ್ರೆ there is mitosis. See, pollen grain first, before it is formed by meiosis, later on it will undergo mitotic division and it produces 2 cells, that is vegetative cell, generative cell. Hagagi mitosis. Then like that mature female gametophyte also, first it is 1 nucleated, 2 nucleated, 4 nucleated and 8 nucleated. Then later on it becomes separated, all the cell will be separated. So that is also by mitosis. Ploidy of the cells. So male gametophyte, male gametes haploid irtavu, hage female gametophyte also will be haploid only. Which advantage offered the seeds? Help angiosperms to adapt for changing environmental conditions. Seeds ಹೇಗೆ ಸರ್ವೈವ್ ಆಗ್ತಾವೆ changing environmental conditions. ಆ seeds ಅಲ್ಲಿ ಏನಾಗ್ತಂದ್ರೆ male female gamete fuse ಆಗೋದ್ರಿಂದ 2 different characters will come and it brings variations so that the seed can survive in a different environmental conditions. So 43rd question. Mendelian disorders may be autosomal or sex-linked, dominant ಅಥವಾ recessive, homozygous or heterozygous, ಯಾವುದಾದ್ರೂ ಇರಬಹುದು. Pedigree symbols ಅನ್ನು ನಾವು ಯೂಸ್ ಮಾಡ್ತೀವಿ to analyze this disorders in the human population. Mention the genotypes for the following pedigree symbols. They have given these symbols, they have given this one. We have to give the pedigree symbols genotypes. Autosomal dominant ಆ ರೀತಿ ಇತ್ತಂದ್ರೆ ನಾವು capital A small a ಆರ ಬರಿಬಹುದು ಅಥವಾ capital A, we can write like this. ಅದೇ autosomal recessive ಇದ್ರೆ small a ನೇ ಬರಿಬೇಕು. ಅದೇ ತರ sex linked dominant ಇದ್ರೆ, sex linked dominant ಇಲ್ಲಿ ಯಾವ್ದು ಕೊಟ್ಟಿದ್ದಾರೆ, like this square ಕೊಟ್ರೆ it is male. So male ಅಲ್ಲಿ XY ಇರುತ್ತೆ. ಸೊ ಡಾಮಿನೆಂಟ್ ಅಂದಾಗ ಇಲ್ಲಿ ಕ್ಯಾಪಿಟಲ್ ಎ ಬರಬೇಕು ಡಾಮಿನೆಂಟ್ ಅಂದಾಗೆ. ಅದೇ ರೆಸೆಸಿವ್ ಇತ್ತಂದ್ರೆ ಅದೇ ಮೇಲ್ ಇದ್ರೆ ಎಕ್ಸ್ ವೈ ಬರೆದು ಸ್ಮಾಲ್ ಎ ಬರಿಬೇಕು, ಫಿಮೇಲ್ ಇದ್ರೆ ಎಕ್ಸ್ ಎಕ್ಸ್ ಬರೆದು ಸ್ಮಾಲ್ ಎ ಬರಿಬೇಕು. ದಿಸ್ ಈಸ್ ಹೌ ಯು ಕ್ಯಾನ್ ರೈಟ್. ಸೊ ದೀಸ್ ಆರ್ ದ genotypes which are used. So last question, justify the below given statement. RNA polymerase cannot transcribe a gene by itself. RNA polymerase cannot transcribe a gene by itself. Other than RNA polymerase alone cannot do that one. So transcription madakagala adu. So what is transcription? It is a method where the DNA produces RNA and the DNA produces RNA by RNA polymerase. Adu onde transcribe madakagala. So because it requires initiation factor and termination factors. Initiation factor like sigma factor, termination factor like rho factor. Transcription and translation are separated by site and not coupled in eukaryotes. Transcription ಮತ್ತು translation both are not coupled same time ಗೆ ಆಗಲ್ಲ eukaryotes ಅಲ್ಲಿ. But in the textbook they have given prokaryotes transcription and translation are coupled because there is no separation of cytoplasm as well as nucleus. But eukaryotes ಅಲ್ಲಿ there is a separation of nucleus, cytoplasm. So in the nucleus there is transcription, in the cytoplasm there is translation. You have to mention that here. So which RNA polymerase enzyme involved in the transcription of 5S rRNA? So here 5S rRNA is produced by RNA polymerase 3. So these are some questions of the model paper 1. Hope I think you have understood well, and if any doubts you have in understanding or any doubts about the answers or questions, you can comment in the comment box and you will get the answer, and prepare well for your exams. All the best.